Hello, everyone. I'm still figuring out why I'm here because I'm actually a reproductive physiologist that just happens to like genetics a lot. So I just recently moved to the University of Wisconsin-Madison, but the work I'm going to present here is some of the work I did at University of Florida as a student and what I did at Mizzou as a professor before moving to Madison. I was asked to talk about protecting breeding populations through genomics. And then I started looking, right, a little bit of literature and more from the conservation and adaptation perspective is talking about understanding and identifying genetic features that make a species adapted to an environment. And I'm using environment in a very loose way, management, the conditions that we give for managing cattle or other livestock. And then I try to connect that to what I was doing. And pretty much we're looking for genes that are present in individuals that help them or variants in those genes to adapt and increase performance in specific conditions. And over time, right, the use of genomic selection as a breeding strategy can help us increase the ability of those animals to perform in those certain conditions. When we think about that genetic selection, I think Dan Janola talked about this yesterday, and where most of the basic scheme that we have used is selection. When we use genomics using, okay, can you see it there? Yes. Single nucleotide polymorphism or SNPs, which are point mutations in the genome. Let's put it in context. If we think about cattle, right, that's roughly 22,000 coding genes, all the same genes, and it's actually those point mutations or those in that combination of mutations that makes an animal be the animal it is. And it's the same for us. We all have the same genes, and it's a combination of all our variants, which give us the characteristics that we have in presenting the environment. So I was looking for the information. This is the length in base pairs of bases throughout the entire bovine genome. And these are the natural occurring point mutations. So a lot. We're not like inventing the wheel here. So there is a lot to work for and that we can use for selection. So the work we have been doing is trying to understand and if we can select animals for reproductive function, increase our knowledge into physiology, and eventually, right, trying to figure out if we, by knowing these genetic variants, we can improve management as well. So we can direct a little bit of the management. And I'm going to focus on Holstein. That's what I've been working in the last few years. If we think about Holstein and the eternal fertility story in Holstein, I think you have seen this graph before. In black is the trend for the breeding value for the genetic merit for fertility. And in pink, the phenotype, what we measure at the farm for fertility in the animals. So the last 50 years, well, no, actually, earlier than the last 50 years, we have a decline in the genetic trend, both and in the phenotypic trait for fertility. It was paired to intense selection for production. And at that moment, we knew those are negatively correlated. Eventually, when we started improving management, also increasing our knowledge on genomics and using genomics, those trends are starting to reverse. Genetics has not catch up yet with how that trend, the phenotypic trend is going up, but a lot of this is management, right? We have improved in the use of technologies. We have improved in the reproductive management that we have at the farm. The one thing that makes it difficult is fertility, like other complex traits, have lower heritability. So we have a lot of environmental component that we see in the phenotype that we measure, right? And just again, to put in context, higher DPR value means that the animal takes less time to get pregnant. It's a reflection of days open. So one question that we had is how can we identify genes involved in reproduction? Most of the work has been done using genome-wide association studies. I think most of you have seen a Manhattan plot. Each one of the color is each one of the chromosomes. And here is just many dots packed. The higher the dot, the higher the association, the chromosome with the trait against which is measured. So we have done that more traditionally with chips, genotyping chips. We know some from clarified, for example, for Zoetis and some other commercials. Another approach is using candidate genes, where we're pretty much trying to figure out markers of point mutations that can be in regulatory sequences of the gene or encoding regions that might affect the protein eventually. And as technology is getting more accessible, we have more and more genome sequence available from animals. So when I think about fertility, why is this so complex? I want to think about how difficult it is to get an animal pregnant, right? It's difficult to get a cow pregnant. We have to have gametes that are intact and of good quality. They have to be able to, the sperm has to be able to fertilize that egg. That egg has to then become an embryo and develop to a blastocyst sometime. If it's by artificial insemination, will stay in the cow. If we do it and produce the embryo in vitro, we need to have a morphologically correct, because we don't really have another option to really use massively in cattle to select embryos, so it's a lot morphologically. We transfer that embryo needs to elongate, needs to signal for maternal recognition. That means that the embryo needs to be growing, the conceptus needs to be elongating, signaling properly, but the cow also needs to be receptive to that. That conceptus later needs to start the process of placentation and then keep the pregnancy. So when I think about that as a physiology, are so many processes. These are just the first 70 days of pregnancy that are going on. So, all of these processes are controlled by many genes. So it's not surprising that you have really that much input from the management environment, that we have a lot of difficulty sometimes to define these phenotypes to be able to increase our ability to find those genes related to each one of these processes, because this is difficult to measure. So we did this is a work that we did in Florida. We started finding and try to curating through different databases and try to find genotypes through the literature, phenotypes, sorry. And then finding markers or SNPs that were present in genes that were either differentially expressed in tissues that were related to reproduction or in different conditions of fertility. For example, they were associated with maturation of the oocyte or the follicle, mammary gland, differentially regulated through the transition period in the liver, uterus, etc. We ended up with like 1,000 genes, and then once we start curating down, we ended up with 77 that were associated with fertility. So the work that I did in specific was trying to validate those SNPs. It was one SNP per gene, so we wanted to have 77 genes, so a small number compared to all the markers that are present in the genome. And we tested those in populations that were either high fertility based on DPR value or low fertility. We did this in about 2,000 animals between Florida and California, and we collected genetic values for fertility traits and phenotypic information from the farms to try to understand what was going on and how we can connect that phenotype and genotype. We then start doing the association. Particularly, we found out of these 77, 39 that were straightly associated with daughter pregnancy rate with fertility, and we try to match that those up with the conventional chip that is using for genomic evaluations and that is released 3 times a year in the United States. So the EUF are those 39 markers. So we compare just with the regular USDA chip, including those 39 markers, and then removing markers that were close to those SNPs that we were tagging to try to understand how much of that genetic variation we were explaining by these SNPs in candidate genes. And we did have an increase in reliability, so the confidence that we have on the data, very small, but I'm just going to remind you, these are only 39 markers. And if we only use those 39 markers, we had around 2.7% of that reliability in the genomics. So very modest, but if we compare to some work that was done by Paul Van Raden in 2013, when they increased by 300,000 markers, they increased 0.4. So by targeting those SNPs, those little markers and genes that we knew that were related to reproduction, we could increase a little bit and at least improve our confidence and our understanding, because we knew that those genes were related to physiology, to reproduction, and not just link. So we can study them a little bit further. When we measure the phenotype, these are expression of the 3 lactations that we could measure in all those animals. You will see that we have fewer animals in the 2nd and 3rd lactation, but it's just because where the animals were in the herd at the moment that we did the measurements. Animals with high fertility, as expected, had higher pregnancy rates. They took fewer services to get pregnant, and they got pregnant faster. And that was true throughout the lactation. So that's a persistent thing that even with more maturity, proliferant pressures of lactation, those animals continue to be more fertile. So meaning, even though the heritability of DPR is low, selecting by DPR does have something that you can measure at a farm, and you can see the improvement in fertility. So when I think about this in terms of conserving the genetics of breeding population, I'm thinking about a functional herd, right? And which are going to be the seed stock for future generations. And you want those animals that have the natural ability to get pregnant and be fertile and continue the populations. The gene that had the higher association in our data set is COQ9. So I'm not going to go through the entire story of COQ9, but it's really what I do want to mention is this gene is really interesting because it's involved in mitochondria. Mitochondrial function, mitochondria is in every cell of the body. Mitochondria is particularly important in the oocyte quality for reproduction. So we had 3 variants, right? So if you have the AA variant at that point, you had a higher pregnancy rate, animals had a higher pregnancy rate, require fewer inseminations to get pregnant and fewer days to get pregnant, as compared to they get the homozygous on the opposite, okay? What's really nice about this gene is when we measure production, because we're always thinking if we select for reproduction, we are selecting against production. That's not necessarily true every time. So in this case, we really didn't have a difference in production or in any of the solids in the milk. So not always selecting for reproduction is bad for production, right? So we need to, we know more now than we knew 10 years ago. And I think we are curating those SNPs to have those databases to have a better selection scheme. We then also check all sites on animals that were having either one of the genotypes, like the good one for fertility, the heterozygous, and the less favorable for fertility. And what's really interesting is the animals that have the good genotype for fertility had increased mitochondria in the oocyte. With at this moment, this in oocytes mature in vitro, which means that they really went through that cytoplasmatic maturation better, and they actually could multiply that mitochondria. So in this case, one SNP, right? I'm not saying this is the SNP that is going to solve fertility, but when we're looking for these candidate genes, we might find some stuff and some markers that can give us some insights in physiology. So again, to put in context, higher oocyte mitochondrial number is associated with increased oocyte viability and quality, which can be important, for example, for donors, for in vitro embryo production, right? Another gene that I just want to touch is Hitchcock protein 1L. We measured the expression. This was also associated with fertility. This is a marker, a SNP that was in the promoter region of the gene. So we expect difference in the amount of protein expression, highly expressed in the oocyte and early embryo, and declines as it goes through. And what is really interesting, this was either you have a cytosine or a deletion at that point. And apparently the embryos, they inherit a deletion, they have higher chances to survive to the blastocyst stage. So what we did is we grabbed embryos that we knew the genotype and we subjected to different stresses. Just to give you a little bit of perspective, we do a lot of these heat stress experiments in vitro to try to mimic a little bit of the stress an animal can experience during summer, for example. So we subject these embryos to different heat shocks, different oxygen conditions just to increase the stress, and we provide them some antioxidants as a palliative for those stresses and measure development. I'm not gonna stay much on this, but just as expected, the red bars are when the embryos were under heat stress conditions. And here you have either without the antioxidant, with the antioxidant, and in different oxygen conditions. But the only thing I want to point here is under heat stress condition, development goes down as expected, right? You decrease embryo production, and which is true also in an animal in the farm. Under heat stress, and I'm from Honduras, so we suffer about that a lot. You can start seeing fertility going like this in the summer, and then start going up later in September, October, when it starts cooling down, at least in the Northern Hemisphere. And these are reactive oxygen species, so whenever we subject that embryo to heat shock, it lights up like a Christmas light because it's just reacting and stress. But what's really interesting here is when we genotype the embryos that made it to the blastocyst stage, the ones that survive the stress, those that inherit either one or the two deletions, right? You have the deletion in one allele or in both, they survive more that heat stress than the ones that didn't have it. So again, if we want to put it in perspective, these can be markers that can be used to try to select those donors, those cystocs that we can use and conserve those populations. One question, and I started doing this at the University of Missouri, is can we find some markers associated with embryonic development itself? And why is this important? We need embryos, right? The animal needs to produce an embryos to maintain a functional herd. Adoption of reproductive biotechnologies. I think when I think about this, if I find a set of markers that is associated with embryo production, those are going to be my target for donors and for those nucleus herds, right, that we're going to use to propagate those herds. So we use a sire model for this because we have more available. So we tested 65 different sires in vitro, measure the ability of those sires to produce embryos that cleave and develop all the way to the blastocyst stage. So here's 1 through the 65 cleavage we use as a measure of fertilization. Was kind of a little bit all over the place. Not surprising. In an in vitro setting, we push the sires, right? We use different additives in the media to push them to fertilize. But what's really interesting is that production of embryos. So we have sires that were very, very good at producing embryos, and we have sires that were very, very bad at producing embryos. And that was consistent across multiple times that we used the sire with different oocyte sources. So there might be something there that was of our interest. So we did a full genome sequence of these animals. Now, due to time, I'm not gonna go through all the methods. But what was interesting, as we refine our phenotype, as we continue to phenotype more sires and genotype them, we start finding some particular signals associated with embryonic development. This analysis was done by Kim Davenport, who's a postdoc with Thomas Spencer and myself. And she just put a few examples here when you have, like, say, 2 copies of the allele that was associated with higher cleavage rate, higher fertilization. 1 copy, in this case, we didn't have homozygous on the opposite. And then we have several genes like that. They have a kind of nice linear effect. So those can be really nice candidates also for selection. And we did that also for blastocyst rate, right? So we have some markers that had a really nice effect, linear effect, and they can be used probably for selection of blastocyst rate. We still need to validate. This is preliminary data, but at least it's encouraging to us that as we improve our phenotypes, if we can start breaking down that pregnancy in little pieces and try to study what are each one of those components, we can start find candidate markers that we could use for selection for fertility. We try to start looking if those markers were near some other ones that were related to fertility. We found some bovine sperm proteins we knew that are associated with fertilization, some lethal, this is a lethal one in Jersey, and some other genes that are important for fertilization as well, and have been associated with other fertility traits. So maybe we can start curating more of those databases of SNPs associated with fertility in this type of traits. And just to conclude, part of what we're doing on our efforts is trying to include markers that can be more informative for fertility, that can be used for genetic selection, and that we can probably substitute some of the less informative markers for some of these. Adapt management, according to that animal genetic merit, there is some nice data from Julio Giordano at Cornell. When he did different reproductive management, the animals with high genetic values for DPR will perform always better, no matter what the reproductive management was. So I think that also speaks to that selection for fertility. From the academia side, we're interested in those functional studies to understand more of the physiology and understand more of that particular management. When I think about where this biotechnology is going, particularly since we have lost some of the homozygotes through those selections, there are a lot of, we're not gonna solve all the genes at once, right? It's not that, but we can start reintroducing genetic engineering, can reintroduce some mutation that probably through selection we lost. And we knew that selection was driven probably by market at the time, right? Well, pain by milk, that's what people was worried. But we might have lost some of those interesting variants for fertility. And now with all these biotechnologies, we have a really opportunity for reintroduction. And with that, I want to thank you and take any questions if you have one.